Good afternoon, ladies and gentlemen. Thank you all for being here. I'm very glad to have this opportunity to share my thoughts about rapid diagnosis of COVID-19 with lateral flow assays based on loop-mediated isothermal amplification biosensors with you. My name is Huang Wenyi from National Taiwan University, and my advisors are Professor Nan-Fu and Professor Lin Qiwan. My submission number is number 87. I've divided my presentation into three parts. I'm going to start with the influences, present situation, and recent diagnosis of SARS-CoV-2. Then I'll talk about my motivation, the idea of immunity passport, and a new alternative of nucleic acid amplification method called LAMP. And finally, what solution we've got now. Let's start with introduction. Compared to SARS-CoV and MERS-CoV, while the fatality rate of SARS-CoV-2 is not the highest, it is the most infectious that it has caused more than 200 million cases. People suffering from fever, difficulty breathing, and other severe symptoms. It has also caused more than 4.7 million deaths, making people more and more nervous about the pandemic. What's worse, the outbreak of several variant viruses make recent antigen antibody point-of-care tests not able to specifically diagnose. Additionally, laboratory diagnosis of nucleic acid is time-consuming and expensive. The efficacy of the vaccines was measured, but effectiveness in the real world is still not to be sure. World faces worst recession since Great Depression after the outbreak of COVID-19. We can see figure at the right bottom. World's annual GDP growth in 2020 was minus 3.59%. So, how does SARS-CoV-2 infect people? There are spike protein, M protein, envelope protein on SARS-CoV-2. Among these, spike protein S1 and S2 binds to the receptor ACE2 in human body. By ACE2, the virus enters our host cell and release virus RNA and start replicating more and more virus to infect our body. There are several symptoms of SARS-CoV-2 infection. We must be aware that some are just like seasonal flu. Recent diagnosis is testing the concentration of IgG and IgN antibody from respiratory swabs, or using PCR to know the CT value. Early identification of infected patients can hurdle its transmission and help controlling the pandemic. However, the development of immunity to a pathogen through natural infection is a multi-step process that typically takes place over 1 to 2 weeks. That is, human body can only generate antibody after infection for about 1 to 2 weeks. PCR can test CT value and calculate specific viral load, but it's too time-consuming and expensive. It is also hard to find asymptomatic patients to conduct a PCR test. Thus, nucleic acid-based point-of-care applications are urgently needed. If you want to go abroad, travelers who arrive at an airport in Taiwan must present a negative COVID-19 RT-PCR test report issued within 72 hours prior to flight. However, there's still a risk of infection in these 72 hours, and the virus will invade to other countries. Therefore, land-based immunity passport can help. It is rapid nucleic acid-based test before boarding. Only takes 30 minutes to conduct. It is a faster and cheaper alternative to PCR. In this study, we used loop-mediated isothermal amplification to amplify SARS-CoV-2 DNA and test it through lateral flow assays to show results. What is loop-mediated isothermal amplification, also called LAMP? Compared to PCR, they are both nucleic acid amplification tests. LAMP is an isothermal amplification method, can be conducted using a simple water bath or a heating block in 65 degrees Celsius, and only for 20 to 30 minutes. While PCR requires a thermocycler, working at 3 temperatures for several cycles, and the reaction time is about 2 hours. Therefore, LAMP is more convenient, faster, cheaper, and more suitable for point-of-care tests. In LAMP, there are 4 to 6 primers specific to identify regions of target DNA or RNA sequence. Amplification initiates from strand invasion by one of the inner primers, and the strand-displacing BST DNA polymerase extends the primer and separates the target DNA duplex. The first product is then displayed by synthetics, initiating from an outer primer, which anneals to an upstream target region. As it is displaced, the end of the product forms a self-hybridizing loop structure due to inclusion of the reverse complementary sequence in the inner primer sequence. This annealing and displacement cycle repeats on the upside end of the target sequence, and the resulting product is a dumbbell-shaped structure that forms a seed for exponential LAMP amplification. Here comes the materials and method in this study. First, we prepare LAMP reaction mix, built-in label primer, BST DNA polymerase, and synthetic SARS-CoV-2 DNA template as our reagent. Then, use a simple heating block to conduct LAMP reaction at 65 degrees Celsius. After incubating for 30 minutes, hybridize FITC labeled probe onto the amplicons for 10 minutes, and stop the reaction at 80 degrees Celsius for 5 minutes. Use pipette to dip final product onto a lateral flow dipstick. The gold nanoparticle will flow through the test line and the control line and visualize the results. If the amplicon exists, it will show 2 red bands, both on the test line and control line, interpreting positive results. If the amplicon doesn't exist, meaning negative result, and we can only see 1 red band on the control line. The last part of my presentation is results and discussion. We designed 3 different probes complementary to the orange sites in the figure to compare probe hybridization affinity. We find out that if labeling biotin at outer primer F3, it shows weak results on the test line. So that we change our design to label biotin at loop primer, and we can see strong results on the test line. Experiments prove that the lateral flow results will show positive if amplicon, biotin, and FITC label probe exist at the same time. However, after conducting lots of experiments, it reveals some challenges we have to be faced with. LAMP's potent amplification mechanism, while making the assay highly sensitive, also renders it highly susceptible to carryover contamination, wherein amplified DNA products from previous LAMP reactions become templates for re-amplification that might lead to false positive results. We can see the figure of the results of GEO that it shows some non-specific amplification on negative controls, which means that there's contamination in the environment of the laboratory due to previous LAMP reaction. Heaps of research shows that opening the reaction vessel during the amplification reaction will increase the risk of reintroducing contaminants from the environment. Now that I dedicate to solving the problem of contamination and non-specific results, so that we are seeking a closed vessel method that can lower the risk of carryover contamination. Using microfluidic system to perform lab on a chip is a good choice. In conclusion, we developed a technique that can be used for SARS-CoV-2 point-of-care test. It shows high sensitivity that the amplification results lead to red bands on lateral flow dipsticks. The target DNA can be amplified in only 30 minutes and can be specifically diagnosed. LAMP-based lateral flow biosensors are of great significance and have high potential for using in clinical diagnosis applications due to the high sensitivity and bioaffinity of lateral flow assay to FITC targeting of the binding sites biotin ligand, and anti-FITC to detect biomolecules. This technique is cheaper, more convenient, faster, and without specific machine compared to other methods. It can assist people to detect the disease earlier and also find people who are potentially infected to avoid asymptomatic infection, infecting others as soon as possible in order to slow down the disease transmission. In the future, we will use clinical samples to test our length-based LFA biosensors. This method can be used as immunity passport, only requires 30 minutes to conduct rapid nucleic acid tests in the airport before boarding. It's also a good idea to combine with microfluidic system to design lab on a chip and can minimize the risk of contaminations and infection to other people. We hope we can contribute to the early detection of COVID-19. Thank you for listening.